Hello and welcome to this special show on Rajya Sabha television, wherein we talks about how India is fighting the COVID-19 pandemic and obviously all aspects of it, be it the digital technology or the scientific ways and means of finding a solution to several problems which have been raised by the COVID-19 disease. Now, today we will be talking about the Sepsivac drug, a kind of immunomodulator which is being worked upon by the CSIR, repurposed rather, and developed along with Cadilla Pharmaceuticals to try and find ways to boost the immunity of those who are in direct touch with the COVID-19 patients or even those who are infected by it with mild symptoms or the ICU patients as well. So we have two important guests with us today who will brief or give us all details about this particular drug, how it works, what kind of expectations are from the clinical trials which have begun in three different parts. So let me first introduce the guests, beginning with Dr. Ram Vishwakarma. He is the director of Indian Institute of Integrated Medicine with CSIR. Welcome, Dr. Vishwakarma, to Rajya Sabha Television. We also have with us Dr. Sarman Singh. He is the director of All India Institute of Medical Sciences, Bhopal. Dr. Singh, welcome to you as well on Rajya Sabha television platform. Let me begin with you, Dr. Vishwakarma, and let's start by trying to understand what exactly are we talking about when we say Sepsivac or immunomodulator.
Yeah, thank you. I will just give you a small background of what we are dealing with this virus with. This is a coronavirus, as you know. This is a new mutant of this, but it is drastically different. Now it is day by day appearing that's drastically different than its previous cousins, Corona 1. And it is appearing that the kind of growth it has is slower than the previous version. It's like it takes 6-7 days for virus to really start manifesting minor pathology, and then once it travels from throat to lung, and then manifestation. And you must be hearing that last 2 days report coming from US that it is going to brain and causing a stroke. So it's a quite different pathogen that we are dealing with here. And all over the world, people are trying to use different approaches, immunotherapeutic approaches or therapeutic approaches. Within the immunotherapeutic approaches, there are three segments one can look at. One is which we use normally, which is prophylactic. When we are born, we are giving these vaccines like BCG, all these that we know, which protect us, gives a memory throughout the lifetime. The second category is that we can modulate the immune system to deal with a particular situation. These are non-specific reagents, but they give you protection against series of pathogens. Okay. For example, BCG. BCG will protect you several pathogens, right? And as you know, BCG trials have also started by US, UK, and Japan. Large trial have started in our own country last week. It started Bombay. BCG trial has also started. The third category is obviously antibodies, which whole work is working to look for a specific neutralizing antibody against this. This product, how we got into this product is under development for many, many years. From 2007, a partnership between Cadilla Pharmaceutical and CSIR under our NIMITLY program, through which we work with industry to take molecule from preclinical, clinical, and then proof of concept to the market. So under that program, this drug was being developed. And around 2012, this got approval for a very deadly disease called gram-negative sepsis. This is a disease which is caused by gram-negative bacteria. And it's a very, very, very difficult disease to treat if you are infected by those kind of gram-negative bacteria. Most of the antibiotics that we use are basically gram-positive. Now, when we got this data and this got market authorization by DCGI, it's an approved drug for one therapeutic indication. And current situation is such that we do not have time to do a new drug development because it is very long process. So then we quickly analyzed 1 month ago with our team and Cadilla team that should we repurpose it. Then we analyzed the biology, particularly the pathology of gram-negative sepsis and pathology of COVID-19. And we found certain features which are common, and then we built a hypothesis that how do we boost the innate immunity and can we deal with this situation? And then quickly, we went to drug controller. Drug controller then gave us first trial approval for phase 2 trial. which is for serious patient, which Dr. Sarman Singh's Institute and All India Institute of Medical Sciences and PGI Chandigarh are involved, and they will take care of that. 2 days after that, DCGI gave us approval for larger trials, a trial of 600 patients for the people who have tested positive, but they are not symptomatic overtly. A trial then later on people who are supposedly resolved, but as we know that the virus lingers on beyond 14 days or 28 days, you must be hearing that governments have now changed the guideline for 28 days isolation. So that is how we came to this situation. And luckily, we have now regulatory approval, and we want to test this hypothesis, whether this can help us in terms of dealing with the symptom, overt symptom, and rescue patients from difficult situations.
Okay. Before I go to Dr. Singh here, one more question on Sepsivac, Dr. Vishwakarma here. What is it that specifically, this particular drug you believe is going to do in terms of those who are either infected with COVID-19 or those in whom you want to, boost immunity specifically? Because as you pointed out, originally this drug was for gram-negative sepsis.
Yeah. So, see, one of the major pathological symptoms which is emerging is lymphopenia in these patients, whether the virus is destroying spleen and other organs of lymphatic system. So this is the one major measurement. Clinicians all over the world are doing this. So idea here is that this is basically a activator of innate immune response. Innate immune response is the one which we are born with. We are born with this set of innate immune equipment you can call it, certain set of mechanism, which protects us throughout our life. Then there is something which we acquire as we grow up to age 20 something, that is acquired immunity, that is antibody driven. Now, innate immunity components that are involved, particularly which recognize certain molecules from the virus. And here, this is basically heat inactivated Mycobacterium W. This is a microorganism which we have killed it, but it contains all the molecule of microorganism, which we are giving IV injections, very small dose, 0.1 ml to 0.3 ml. Very tiny dose we are giving and boosting the specific components of innate immunity, so that there is an internal mechanism of body which can clear this virus and stop the replication of the virus.
Okay. Let me bring in Dr. Sarman Singh here, the director of AIIMS Bhopal. Dr. Singh, as Dr. Vishwakarma is pointing out that yours is one of the institutes where the clinical trial of phase 1, as far as the serious patients are concerned, infected with COVID-19, of Sepsivac drug is, going to take place. So your views on Sepsivac as an immunomodulator, and then about the trials as well. How are you going to go ahead with it? What are the protocols? How do you plan to go ahead with it?
All right. So as Dr. Vishwakarma very rightly said that as of now, we know that, thousands and thousands of the people are dying because of COVID-19. And right now, he very rightly said, and I will reiterate that right now we don't have any antiviral or any vaccine. And therefore, we don't have any option but to, repurpose. Repurpose means the drugs or whatsoever, a vaccine or anything which is for other diseases, they can be used for another disease. So they are known as repurposing. And we know that hydroxyquinoline or any other antiviral which are used for HIV, or there are other drugs, they have also been repurposed. Similarly, Cadilla and Dr. Vishwakarma's institute, they came with the idea about this Sepsivac. Now, let me tell you that Sepsivac is what Dr. Vishwakarma told. I would like to give a little more introduction about this. Before the trade name Sepsivac came into Mycobacterium W, this was in fact is a mycobacterium which is saprophytic mycobacteria. The one mycobacteria which causes disease, that is tuberculosis. This is another one which doesn't cause any disease, and it is found in the soil and water. And it was 1978 when Dr. G.P. Talwar at AIIMS, New Delhi, he found out that this is a very important mycobacterium which can stimulate the T cells and proliferation of the T cell. That's what he was talking. And then it can have the T memory cells and lymphocytosis. And that was the reason that in 1990 for the first time he started clinical trial of this Mycobacterium W for leprosy, because this mycobacterium was isolated from the soil and the areas of the leprosy patients. So he thought that probably some of the patients, those who are not developing leprosy, about 2% or so, even though they are residing in the family of the leprosy patient, they are not developing leprosy. So what is the reason? So probably he thought, and this idea came to his mind, and then he gave this injection of this Mycobacterium W, which is of course now changed. Name is changed. Now it is Mycobacterium indicus pranayi. So MIP, MIP. So then this trial started in 1990, continued for many years, 2000 up to 2005, and then larger trials they were carried out, and they found that yes, this Mycobacterium tuberculosis, if it is heat killed and given intradermally to the patients of leprosy, it can definitely make change. And many of the leprosy patients, they can be cured, or their morbidity, means deformation of the disease, can be changed. Later on, this, as he very rightly said, that this was, technology were transferred to Cadilla. But in the meantime, this was also tried after the Cadilla took it. Then these injection or this immunomodulator, this was also tried in the bladder cancer, also in lung cancer, also in the genital warts. So that is, this is, they have been trying, and they thought that probably on the same lines where they have seen that, it causes T immune cell stimulation and proliferation, lymphocytosis. It enhances our immunity, that is non-specific, but also we presume that probably other than innate or non-specific immunity, probably this might also enhance some T memory cells. And these T memory cells, which remain for a long time, even though coronavirus is not a very chronic disease, but it is presumed that probably this might help to develop the antibody production also, specific antibody production. So now we are also part of that, as he very rightly said, that there are 3 institutes, that is AIIMS Delhi, AIIMS Bhopal, of course, and then PGI Chandigarh. So we will be recruiting patients from tomorrow. We are getting this medicine. And this is, of course, all valid, documentation and everything, permissions and approvals. We have received it. So from day after tomorrow, probably we will be starting it. To begin with, how the trial will be done, because as we know that it is a good thing, I must say that in coronavirus or COVID, not more than 3 to 4% they will go to ICU, means critical. Okay. And the fatality is very low. This is a good thing, but still, by any mean, even in the red zone, which we are in Bhopal area, we will not have more than 8 or 9% of the patients who will require oxygen. So now, obviously, once the patient goes for oxygen requirement, that is considered to be the serious patient. Otherwise, if they are not having any other comorbidity, they are adults and healthy, then usually they will have minor or no symptom at all. So 80 to 90% they will not have any symptom or very mild symptoms. Only about highest number is reported is about 10% they will require oxygen. And oxygen, some of them they will require oxygen by just tubing outside, and very few, about 3 to 4%, they will require what we see tracheostomy, means you have to cut the throat and then put the tubing. So that they are considered to be more serious.
For these 8 to 10% patients?
Exactly. To begin with. So this one will be this group. There are 3 groups we have we will be doing. So one group, obviously, the first group will be this one. So critically sick patients, they will be injected 3 injections. So 1 injection daily, a very tiny dose, about 3.3 ml. And then we will see on daily basis, and then we will be monitoring laboratory that whether lymphocytes are increasing and the patient oxygen supply, because the oxygen supply is important. So we have to see that, say, for example, requirement of the patient was not meeting earlier, how quickly he can, take breathing on his own. So oxygen requirement, say, for example, earlier it was 60% requirement or 80% requirement. If we can reduce 50% or 40%, so less than 50%, obviously, we can say that, he can be extubated or something like that. Okay. And then how many of these critical patients, they survive and they come out of the critical care, and then, so that will be one group.
Okay.
The second group is, of course, where we will see that, how quickly, if they are hospitalized, but they are not critical, but how quickly they can convert from the corona positive to corona negative. So
Okay.
that will be
how quickly they recover?
how quickly. Usually it takes about, so far our experience is that it takes about 8 to 10 days. So say, for example, if they can recover in 3 or 4, 5 days, burden on the hospital, psychological pressure or stress on the patient, all will be obviously much desirable. Mm-hmm. And of course, the third group also we have included in our center, which we will see that what happens in the healthcare workers also if they are given. That will also see.
Okay, and how much time, Dr. Singh, you believe this trial or the study will go on before we get to know the results? How long are you going to conduct this?
Let me tell you, you will be surprised. It is a very fast study. So in about 3 to 4 weeks, we will have results.
Okay, that's quite good and quite fast as well. Dr. Vishwakarma, I'd like to bring you in here. Since we are looking at these two positive aspects, one, that the trial process, the duration is not that long, around 3 to 4 weeks. Two, it is not a new drug. It is a drug which is already there. It has been repurposed by you guys. So does these 2 points make it a very effective solution for the time being till we have the vaccine? Or are there some other aspects also being looked at, because Sepsivac might be just one of them?
Yeah, absolutely. So the biggest hurdle for drug to bring from lab to clinic, 2 biggest hurdle are regulatory approval. Mm-hmm. Normally, to develop a drug, it takes 10 to 12 years in the best of the situation. So when Dr. Singh was explaining Professor Talwar's work, imagine how long it took him is 30, 40, 50 years of the work. So it's a very long journey. And so one great advantage we have with this, we have regulatory approval. So even when the early indication from Dr. Singh's hospital come, because of law, that doctors are authorized to use off-label use of a particular existing medication. Mm-hmm. They can quickly start using this in real-time situation. That's advantage we have. Mm-hmm. Even when we are doing the large phase 3 trial, for which we have approval. Number 2, this drug is already manufactured in a GMP facility because this is IV drug. IV drug manufacturing requires a very sophisticated setup. Not many companies have facility to manufacture these drugs. Mm-hmm. So this is already GMP manufactured. Otherwise, it will take, like you must be hearing, people are trying to do new antibodies and still companies have started building up facilities to manufacture them. Here we have facility ready, drug is available. Like tomorrow, drug will be delivered to Dr. Singh's hospital. So these are the 2 great advantages we have here with basically proving this particular point here, that whether this drug is giving advantage to patients, either patients who are critically ill, like he said, or if we can prevent people from going from a normal symptomatic, lesser disease situation, so that we can protect our medical staff. That's mm-hmm. That dimension is very important. Lesser people going to ICU means our doctors and nurses are protected.
Okay, so it is a very interesting situation out here and something which we have at hand. Dr. Singh, I'd like to bring you here, Dr. Sarman Singh, on the possibilities of this particular drug, that is Sepsivac. Now, there are clinical trials which you will start conducting. In terms of the possibilities which it will have in our fight against COVID-19, and from a medical practitioner's point of view, is this our best shot so far?
Yeah, difficult question, but of course, one has to be optimistic. So I would not commit anything, but definitely I am very optimistic, because not only because I am doing the clinical trial, but of course, because there is nothing better than this available. So I think this can be the best available repurposed thing, because hydroxyquinoline also, it has so many side effects. Many, if you are having ECG changes, obviously the patient can die. The HIV drugs, they were also tried, but in China they showed some effect, but the larger study they found that they are not effective. So right now we don't have anything. So say, for example, if something is 0, even if we have 50%, obviously we are win-win situation. We are definitely better. So that is definitely, optimism has to be there. And because, as I said, till any vaccine or any specific drug comes, obviously we might be having, this is the only option. So probably we can give, and this is, as he very rightly said, that it is made in India, number one. It is made in a good laboratory production house, and this can be made available. Thousands and thousands and millions of the patients, they can be vaccinated, or they can be injected. So that all these things for Indians, I can say that it is a moment for cheer if the things they come in in our side.
Okay, that's quite an optimistic statement out there coming in from Dr. Singh, who's going to oversee the clinical trials for the next few weeks. One last question from you, Dr. Vishwakarma, in terms of Sepsivac is one option which you guys have worked upon in the past, few days. Is there something else, your institute or your researchers, scientists will be working on, or something which you are aware on? Because this is a global fight against COVID-19. All, entire scientific community all over the world is trying to find one solution or the other, be it in terms of immunology or in terms of therapeutic terms.
Yeah, so we have two other approaches across the CSIR. One major approach that we have taken, and work is happening at least 5-6 CSIR labs right now for last 1 month very intensively, is that we are developing synthetic processes of close to 15 drugs which are at different stage of clinical development across the world. Now, if a drug today gets approved, for example, Remdesivir gets approved by Gilead in US, it's a good news. But how our Indian population will get it? It is going to be $1,000 per dose. Now, how many of are able to afford it? So what we have done, drug like Favipiravir, Remdesivir, all these drugs which are there, we have developed synthetic processes. We have tied up industry partners, large industry, and we are ready any day to produce kilogram and hundreds of kilograms of those drugs, GMP grade drug for our population. That work we are doing. Second project, very interesting project, you will hear in a couple of days' time. We have been working with another major pharmaceutical company, I will not name it, where we have identified a very specific medicinal plant and its compound.
Okay.
We were working on this plant for some other virus for almost 5-6 years. Another RNA positive RNA virus. Okay, all these viruses, their entry point to the cells are different, but once they go into the cell, their processing by endocytosis and other organs are more or less same. So this substance we have been testing for other RNA viruses, and we have just tested this for this virus also in preclinical situation. And we have gone to DCGI, and we are hopeful that in the next few days we will have some regulatory authorization. So that will also go to phase 2 trial. We already have completed phase 1 trial on healthy human volunteer to see its safety, and this will go to, so these are the two other arms that we are working. Third thing we are working is, as you must be hearing, Dr. Sir will know, zinc is very critical for actually slowing down the virus. There is a full mechanism. So we are working on a specialized complex of zinc, which has 4 times better than inorganic zinc bioavailability. And very soon we will have this parallel trial going on the zinc supplementation along with the therapeutic or immunotherapeutic that you are using. So we are looking at these 3 other options at CSIR, and I'm sure other scientific agencies are looking at this. These are desperate times, let me tell you, and these are the testing time for scientists. The whole world scientific community, I can tell you with firm belief, solutions will only come from science and medical community. So hats off to these two, particularly our medical communities. They are fighting double war. They are fighting war with virus, and they are fighting war with the lack of equipment and supply and support system. I can assure you on behalf of scientific community, world scientific community and medical community, that solutions will come from effort that are happening globally in India and elsewhere. I think the solution to coronavirus are going to come from science, and of course the measures to until a vaccine or drug comes up, measures to kind of measure that we are putting in the society. So I'm very optimistic with the what is happening world over in these areas. A lot of solutions are there in sight.
Okay, definitely. Rightly pointed out by Dr. Vishwakarma and Dr. Singh there as well in terms of the fight against COVID-19. It is a scientific community which is working really hard on trying to find solutions. And as Dr. Vishwakarma pointed out, it's not only Sepsivac, but there are three other processes which are being worked upon by the CSIR scientists to try and find a cure, a solution to COVID-19 virus as to how to fight it. But as far as Sepsivac is concerned, those are the details in terms of how it will enhance the immune system, the defense mechanism of the body, or the innate immunity. And the clinical trials, as Dr. Singh pointed out, will begin at all these three hospitals, that is PGIMER Chandigarh, AIIMS Bhopal, and AIIMS Delhi as well. And it will not take long. It will be only 3 to 4 weeks before we get to know as to whether or not this is successful. All those who are involved with this process are optimistic about it. Let's also stay optimistic and wait for the results to come in. Meanwhile, you and me, all of us, need to follow the guidelines as far as lockdown, social distancing norms are concerned. Stay home, stay safe, and keep watching Rajya Sabha Television.